I request Dr. K. Johnson Ariklasamy to occupy the dais. A beautiful smile is the best universal welcome. It can make the day a good easily. A warm welcome to one and all present here.
Now I request the moderator for the first session of today's program, Dr. A. Joseph Tatayas, Associate Professor and Head of PG and Research Department of Zoology, the American College, Madurai, to occupy the seat on the dais. Now, I do request the chief guest of today's program, Dr. M. Muniraj, to take the seat on dais. Today is the fourth day of the nation's Science Week celebration, the Vigyan Sarvatra Pujyate. The theme for today's program is Swadeshi Paramparik Vigyan and Pradyogiki, the inventions and innovations.
Now, we like to honor our speaker for this session with a shawl and a memento.
And I request Dr. John Ariklasamy to honor the chief guest with a shawl, followed by Dr. G. Joseph Tatayas, sir, Head of the UG Department of Zoology, with a memento.
Thank you, sir. Now, I hand over the session to the moderator.
Good morning, one and all present here. And it's a pleasure for me to welcome all of you for this Science Week celebrations, commemorating the 75th Independence Day celebrations of our nation. So, in the Holy Bible, Psalms 136:21 says, "And gave their land for an heritage, for his mercy endureth forever." So God has given an independent India. So we are cherishing our memories, and we are enjoying the freedom. And the government of India has selected our college as one of the centers for celebrating this among the 75 centers spread all over the country. So our college is also known for the freedom. So we are enjoying the freedom. If you go to other institutions, you can understand how much freedom we are enjoying as a student or as a faculty member. So Deuteronomy 32:7 also says, "Remember the days of old, consider the years of many generations. Ask thy father, and he will show thee, thy elders, and they will tell thee." So it is fitting to cherish our memories and what our forefathers have done, how they fought for the freedom, how they struggled to achieve the freedom, and after achieving the independence, how they excelled in science and technology. So commemorating all these things, we are having this Science Week festival. So in this, I am happy to introduce the speaker of today's session, Dr. M. Muniraj, Scientist D, ICMR VCRC Field Unit, Madurai. So VCRC means Vector Control Research Center, and the headquarters is at Pondicherry, and we have a field station at Madurai. Initially, it was a Center for Research in Medical Entomology, and now it is the field station of VCRC. So our college, especially the zoology department, has so much of association with the field station VCRC Madurai unit. So right from the days of Dr. Ruben, So when she was heading the institute, and our professors used to go there as external experts. Even I myself went twice as an expert, and we had a collaboration with the help of Dr. Selvaraj Pandian, Dr. Soma Sundaram, and even now we have two staff who already worked in the same unit. And our alumni they are also working as scientists, especially Dr. Paramasivam, and earlier Dr. Arunachalam worked there, and even Dr. Tenmoli was a PhD student, Dr. Mariyappan. So all these are alumni of our zoology department of our college. So it is a honor for our college also to receive him as the chief guest for today. And I'm going to tell a few points about his achievements. Dr. Muniraj, as a gold medalist for securing he has received the gold medal for securing university first rank from Madurai Kamaraj University in 1994, and he has started his career as assistant professor. In 2001, he had been selected as Scientist B by Indian Council of Medical Research and posted at RMRIMS, Patna, and then transferred to the now the field station VCRC at Madurai. Now he is continuing his research activities as Scientist D. His research works concentrate on insect vector and vector-borne diseases, especially visceral leishmaniasis, that is Kala-Azar. He has guided around 50 students from different universities of India and imparted training. He has published around 35 research papers in national and international high impact factor journals and written chapters in nearly 7 books. He got 2 patents. He has developed several novel techniques to identify and cultivate vector-borne pathogens. He has received a few awards such as International Einstein Award and Bharat Jyoti Award. He is also acting as reviewer and technical editor for a few prestigious national and international journals and member of doctoral committee and board of studies of universities. So with this short introduction, I now hand over the session to Dr. Muniraj, who will be delivering his lecture on traditional scientific knowledge of our ancestors, the forgotten glory. Thank you.
Good morning to all. I first of all thank Dr. M. Dominic Christopher, the principal of this college, and Deputy Chairman G.C. Abraham, sir, and Finance Officer Dorothy Sheila, and Coordinator John Atikalaswamy, and Deputy Coordinator Priyadarshini, and Dr. Joseph, sir, for inviting me to deliver talk during this auspicious day. The topic which was given to me is Swadeshi, Paramparik Vigyan and Pradyogiki. That means அவருடைய நம் பாரம்பரிய அறிவியல் அறிவு அண்டு அதனுடைய தொழில்நுட்பங்கள் அதை பற்றி பேச சொல்லியிருக்காங்க வித் இன்வென்ஷன்ஸ் அண்ட் இன்னோவேஷன்ஸ். ஸோ I will talk about the major things in the vector control research by touching the areas of our ancestral techniques, Paramparic Vignan. I am starting from a historical note. Everyone knows about the Alexander the Great. He was a king of country Mesopotamia. He came to India. He came up to India by conquering all the countries he passed by with only aim of reaching India, because India was such a wealthy country. During those days, the dream of every Europeans and the dream of the people lived everywhere in the world, they wanted to see once at least India. So that much the glory of India was there. Mainly the glory of India was taken to the Western countries based on the writings of Marco Polo, Megasthenes, who has written lot of things about India. Particularly Megasthenes written about even the Pandya kingdom. In his writing, he has described about the king of Pandya. He has mentioned that king of Pandya ruled most part of southern part of India, and the people were very wealthy in Madurai. This was the sentence of Megasthenes. He has written, Megasthenes, Marco Polo, he has written about Madurai and the wealth of Indian people. Okay. So I will also explain here and there in Tamil also to understand all the students. So in the writings of Megasthenes, நம்ம படிச்சிருப்போம் சின்ன கிளாஸஸில். மெகஸ்தனிஸ் எழுதிய நூல் இண்டிகான்னு சொல்லி சின்ன கிளாஸஸில் படிச்சிருப்போம். அந்த இண்டிகாவில் ஃபுல்லாக they have described about India. இந்தியானா என்ன, இந்தியாவில் என்ன வெல்த் இருக்குது, இந்தியன் பீப்புள்ஸ் எவ்வளவு வந்து செல்வ செழிப்போடு இருந்தாங்க, இந்தியாவில் எவ்வளோ கல்ச்சர்ஸ் எவ்வளோ எப்படி இருந்தது, அதை பற்றி இண்டிகாவில் டிஸ்கிரைப் பண்ணியிருக்காங்க. ஸோ ஆஃப்டர் ஹியரிங் ஆல் தீஸ் திங்ஸ், Alexander got interest towards India. So he started to, along with his forces, he tried to invade India. But at the point of entry to into the India, the Sindhu river was there, and he was facing a terrible fight with the Porus king. Porus king கூட ஒரு பயங்கரமான ஃபைட்டில் ஈடுபட்டு, then even though the Porus was defeated in the fight, but Alexander was, the forces of Alexander was not ready to go further due to lot of losses they faced so far due to the long travel. So they wanted to return back to their home country. So Alexander வந்து அந்த போரஸ் மன்னரை ரிலீஸ் பண்ணிடுறாரு. ஸோ நீங்கள் போயிடலாம் அப்படின்னு. ஸோ அதோட அலெக்சாண்டருடைய கனவு, இந்தியாவை பற்றின கனவு வந்து முடிஞ்சிருது. ஸோ they returned to their homeland. On the way of return, he died. Alexander died. அதுதான் நமக்கு இங்கே முக்கியம். But that I will come at later part. So Alexander return ஆயிடுறாரு. இது மாதிரி later, after hearing the news of India, for example, the Bodhidharma from India, he went to China. He was the first person taken Buddhism from India to China. So just I want to say all these things. You have to imagine the power of India. India அப்படிங்கிற கண்ட்ரினால வேர்ல்டோட ஹிஸ்ட்ரியே மாதிரி இருக்கு. ஒவ்வொருடைய அந்த வேர்ல்டு ஹிஸ்ட்ரியினுடைய ஒவ்வொரு பிளேசஸ்லையும் the role of India is there. So you want to understand, I want you to understand these things. So Bodhidharma went China. நம்ம போதிதர்மா பற்றி நமக்கு அவ்வளோவா தெரியாது. till the சூர்யா ஃபில்ம் வர்ற வரைக்கும் we are not knowing about Bodhidharma. But the real thing is Bodhidharma was a god in China. People are still keeping him as a god and praying in China because he was the person taken Buddhism from India to China. As well as he was the person who taught them how to fight. So, in the Chinese படம்லாம் பார்க்குறோம்ல நம்ம kung fu போடுறாங்க karate போடுறாங்க everything came from India but we forgotten everything. So அந்த kung fu Shaolin சொல்லுவாங்க Shaolin kung fu was taught by him to the people of China. So he taught not only that fighting, he taught so many things, scientific things, because these things learned from Siddhas of Tamils. Actually, Bodhidharma is a young prince of Pallava kingdom. He was from Kanchipuram. So he went to China to talk about the Buddha, and he preached about Buddha in China. After that, but in later part, once the people were something happened, he wanted to return back to India. On the way of return, he means he மறைஞ்சிறார் அப்படியே காத்துல. So still that story is in mystery. After that, after Bodhidharma, 2-3 centuries later, Yuan Shuang came to India to learn about the wealth of India, to learn about Buddha. He came to India, Yuan Shuang. So Yuan Shuang India வந்துட்டு he came up to Kanchipuram. காஞ்சிபுரம் வரைக்கும் வர்றாரு. காஞ்சிபுரம், மதுரை, these are all the famous Buddhist Samana Buddha மதங்களின் and Chinism was very popular in India, in Tamil Nadu, particularly in Madurai. Madurai is having a lot of role in history related with this Buddhism and Chinism. So, so this Bodhidharma has taken the knowledge from Siddhas. Siddhas are nothing but they are scientists of that period. Now we are calling them as Siddhas. ஏதோ தாடி வச்சுட்டு காவி ட்ரெஸ் போட்டுக்கிட்டு பைத்தியமாக சுற்றிட்டு இருப்பாங்க. பட் தே ஆர் ரியலி ஆக்சுவலி அப்போல்லாம் டெசிக்னேஷன் கிடையாது. சயின்டிஸ்ட் ஏபிசிடி எல்லாம் கிடையாது, ஐசிஎம்ஆர் கிடையாது, சிஎஸ்சிஆர்லாம் கிடையாது. ஸோ தே ஆர் ஆல் தி சயின்டிஸ்ட். They are not paid for it, but they are the actual real scientists. They worked, they always thought about the real thing, truth, they always wanted to know in everything. So, the Bodhidharma got these knowledges from the Siddhas and transferred this knowledge to the China. So, China got flourished by the knowledge given by the Bodhidharma. Then later, to understand, I told you, later, to understand all these things, Yuan Shuang came to India. He visited all the places and he has written, he is actually a traveler, monk and traveler. He has recorded everything about India, like Marco Polo's and Megasthenes write-ups. He has written everything about India. And he came up to Kanchipuram and he has written about the Pallava kingdom in Kanchipuram and the way the Buddhism is followed here. Everything he has written in his write-up. So all these things spread all over the world. Particularly all over the world means there were no America that time. So that time only Europe was known to everyone. So the people of Europe got very much interested about India. Even before the time of Alexander, everyone was interested to at least see India once in their lifetime, to see how people are living there, how much richer, wealthier people are living in India, how the culture is wonderful, beautiful in India. Everyone wanted to see at once in their lifetime. So, the European kingdom of that period started, they wanted to see India, they wanted to have trade with India. They used to have the land way, Silk Route and Spice Route it is called. They used to go through that. It reaches through, it goes through the Constantinople city. At one part of history, there was some sort of conflict between kingdoms. They blocked the way in Constantinople to reach India. So they were not able to reach India. Even by land route, it was a very toughest job to reach India those period. Even if 100 people are started to started from, for example, from France to India to see India, only one or two will reach India. So that much toughest route it was. So they used to come all the way from all their countries of Europe to see India. So that way, spice route was blocked due to some political conflict. So அது க்ளோஸ் பண்ணனால என்ன பண்ணாங்க, they wanted to see some another route to see India, to reach India. அதனால என்ன பண்ணாங்க அப்படின்னா, Portuguese they started first. Portuguese they started first to explore the sea route to reach India. So அதனால they started sailing towards India. அதில் first மெயினான வாஸ்கோ ட காமா, he used to sail the crossing the Africa and the Cape Town he crossed, and he reached finally the Kerala, Calicut of Kerala. At the same time, other countries, Dutch and English people, they wanted to have alternate route to this, because so after finding this route to India, Portuguese were dominating this route. They will not allow other countries to travel this route to reach India. So every other country, they were in a situation to find alternative route to reach India. So apart from this Vasco da Gama, other fellows like Magellan and Magellan started to find another route, and Columbus was also want to see India through. Columbus was thinking that India is nearby. Before Columbus period, the European people were thinking that the world is flat one. If people are traveling beyond certain limit, they will fall down from the sea. So they were thinking that the world is flat one. So they were afraid of going to west side to reach India. But only this person, Magellan, sorry, not Magellan, not Vasco da Gama, Columbus was moving eastward to see India, and he was calculating that India will be nearly some 5,000 kilometers away from the European mainland. So he, by very toughest sailing, he crossed the ocean Atlantic and reached a landmass, and he called it as India. Even after his return, until death, he was believing that it was India. But it was not actually India. He found a new landmass that we are now calling it America. So, and that name, your college is also Americancollege is there. So he found new. So everything comes around India. So he found a new route, and he was trying to find new route to India. On that process, he discovered America. So after that, that American area was occupied by the kingdom of the Columbus belongs to country. Then again, Magellan want to see. Magellan want to find. So somebody was not sure that this is India. So they wanted to find route to India. So Magellan started to travel towards west to reach India. And he was also miscalculating the distance, but at the time, he knew that there was a big landmass, but that was not actually America. That was not actually India. So he traveled a long way, and I think out of some 10 ships, they started travel. Finally, they reached 2 ships only, reached the end. They reached Philippines. Some islands in Philippines, they landed. And then the main duty of the Magellan was preaching about Christianity to the people, local people of that island. And one king, I forgotten the name, that king was very much favoring the Magellan, and the people along with himself, like some 10,000 people converted to Christianity there. At the same time, there was another island. That king was not ready to accept Christianity. So there was a fight between the McClellan group and that local king, and finally McClellan was killed without knowing what he discovered. He was the only person shown to the world that the world is a circular one. One can go through even western side to reach India, apart from eastern route. Why I am saying all these things? Because the glory of India, the importance of India in the history of world. And I also wanted to say a few words about our own Tamil culture, Tamil literature. If you take Madurai Kanji படிச்சு பார்த்தீங்கன்னா you can see about the beauty of Madurai. மதுரையை பற்றி எவ்வளோ அழகாக டிஸ்கிரைப் பண்ணியிருப்பாங்கன்னா, மதுரையில் வந்து 6 மாதிரி அகன்ற தெரு இருந்ததா, வானளவு உயர்ந்த கட்டடங்கள் இருந்தது, பில்டிங்ஸ், ஹவுசஸ். அதில் மக்கள் வந்து வீதிகளில் எந்நேரமும் போய்கிட்டு வந்துக்கிட்டு இருந்தாங்களாம், நாலங்காடி அல்லங்காடி அப்படின்னு. மதுரையில் வந்து முதல் சாமம், 2ஆம் சாமம், 3ஆம் சாமம் வரைக்கும் ஹோட்டல்ஸ் நடந்திருக்கு அந்த காலத்திலேயே. பிஃபோர் 3000 5000 years back, which was written in Madurai Kanji. Day and night streets were there, people were roaming around all the places. The people were speaking different languages in Madurai. So, the people from Greek, so many people were there in Madurai. And Madurai seemed to be always a festival city. Lot of festivals were going on, not from this time. It was from very prehistoric period it was going on. Similarly, if you take the Pattinapalai, it describes about the beauty of Pumbuhar, Kaveri Pumbottinam. It describes about Pumbuhar. Pumbuharla வந்து there are several thousand ships were landed in the port of Pumbuhar, and so many traders from several countries, they used to come to the port, and people of this side go to the ship. So many exchange activities were going on. The city was very busy during daytime, nighttime. So many things were described in Pattinapalai. So that much wealthy people our country is, that much wealthy country is ours. But now if you see these days, people are making road, immediately that road is vanished after one rain. There is no proper drainage system. Whenever you go, only they just make road. There is no, they are not thinking about how the rainwater will go. After making road, only they think that, oh, rainwater has to go somewhere. So it will go, rainwater will find some route to go downside area. So that is the engineering capability that these engineers of these present days are doing. Okay, come to this story. So this king, Alexander, on his way return to his home country, was killed. Not by any king, he was killed by a mosquito. He was killed by malaria. So he got malaria, and he was suffering from fever for a few days, and the doctors of his period were trying to treat him, but they were not, it was not successful, and finally he died. And in some history book it was written that he told the followers that his hands should be kept outside to see the people that even though he was a king of that period world, and he was not taking anything in his hand. So that knowledge was given by mosquito to him. So come to our subject. Mosquito is the world's deadliest animal. This is the data of some 10 years back, but even then it was killing 725,000 people in a year, and which was next to human. Human is also an animal when killing other human. So human is also killing another human. Around 4,75,000 humans are killed by human activities, and the rest, the wild animals, the dangerous wild animals are coming only next to human, like lions, tigers, crocodiles, everything comes next to human. These are the major mosquitoes responsible for the transmission of diseases to human. Aedes aegypti, and Aedes albopictus, Anopheles gambiae, and Culex quinquefasciatus. Now, these days, people are not giving much attention to the vector-borne diseases due to COVID. Everyone is giving now attention to COVID-19, and people forgotten to think about vector-borne diseases, which was causing equal mortality and morbidity to the people, but in a slow way. COVID was coming like a tsunami, but this is like a slow poison. It is always there, killing people, and we are not bothering much about this. Major vector-borne diseases are dengue, chikungunya fever, Zika, Japanese encephalitis, filariasis, malaria, leishmaniasis. Because these are all the timings of diseases. Once people were afraid of plague, everyone were talking about plague, and sometimes people were afraid of AIDS. So government was giving more fund for AIDS research, and people were afraid of HIV. Later, slowly it has gone. Now people are not that much afraid, and government is also not that much serious about AIDS, because now it is known that it can be a controllable one. It is under control, HIV. But similarly, these vector-borne diseases are very difficult to handle, because it is not the responsibility of human to human. In between, and another individual is there, that is mosquito. Mosquito transmits disease from one human to another human. We can teach human how to practice those things to avoid mosquito bites, but we cannot teach mosquitoes to go to any house, don't bite anyone like that. So it is difficult. We have to give much attention to this when comparing with other diseases, because in between this vector is available. This vector playing a role. VCRC, after merging the Center for Research in Medical Entomology in Madurai, it was merged with the Vector Control Research Center, VCRC. I have seen the first translation of vector at VCRC. No ஈனி கடத்தி. புரியுமா உங்களுக்கு யாருக்காவது? வெக்டார்னா கூட ஈஸி அண்டர்ஸ்டாண்ட் பண்ணிடலாம் போல. தமிழில் அதுக்கு நோ ஈனி கடத்தி. ஸோ நோயை கொடுக்கக்கூடிய டிசீஸ் காசிங் ஏஜென்ட்டை டிரான்ஸ்ஃபர் பண்ணக்கூடிய ஏஜென்ட். நோ ஈனி கடத்தி. வெக்டாருங்கிறதுக்கு மீனிங் உங்களுக்கு புரிஞ்சிருக்கும். Vector is a vehicle. Vector means it's a transport vehicle that will transmit a pathogen from one person to another person. So we are doing research with that vectors. So our center name is Vector Control Research Center. So we are working with all these diseases. Why it is difficult to control insects, particularly mosquitoes? Because of development of insecticide resistance in mosquitoes. This maybe seems to be a very simple sentence, that insecticide resistance is everywhere. We are giving antibiotic, we are developing resistance to antibiotic. So resistance is not a matter. So you may think like that. Resistance is a simple thing. But when it comes to vector control, development of resistance in mosquitoes is the prime important area for us to study, because the only thing which we are having in our hand is to control vectors. We cannot educate mosquito not to bite, but we can kill mosquitoes by the way of various methods that we have developed and already developed, like chemical methods. Still lot of things are speaking about lot of other control methods, but still the chemical based methods are the viable ones still. But what happens when the concentration of chemicals used for control of mosquitoes, if it is increased beyond certain level, that will be harmful to the environment itself and the human itself. So புரியுதுலப்பா, அந்த கான்சன்ட்ரேஷன் அதிகமாக நம்ம போடும்போது அது நமக்கே வந்து பாதகமாக முடியும். It will kill ourselves. So that was first described by a lady in America through a book called Silent Spring. In that book, he described the impact of DDT usage, uncontrolled DDT usage. That time, nowhere knowing about the impact of DDT. She was sitting in one village of outside outside main cities of America. Somewhere she was sitting in a park, and she observed that lot of birds were falling, and they immediately died. And then people started to investigate about the birds' death, and they found that even the eggs laid by the birds were not so healthy. It was like fragile. The eggs laid by the birds were fragile. Like நம்ம தமிழ்ல சொல்லும்ல, தோல் என்னது தோல் முட்டைன்னு சொல்லுவாங்க. அந்த சாஃப்டாக இருக்கும் முட்டையை நம்ம எடுத்தாலே அமுக்குனா உள்ள புசுக்குன்னு இருக்கும். So such kind of eggs were laid by those birds. So then later, when they investigate the cause for such type of things, they found that DDT was responsible. At the time in America, they were not using DDT for the control of mosquitoes. They were using DDT for agriculture purpose, to kill the insects which are causing diseases to the plants. So they used DDT for agriculture purpose. So you may know that biological magnification, DDT cannot be easily eliminated from the environment. It will accumulate. It will accumulate slowly. Say for example, if it reaches the water body, one fish may take 1 unit of small fish. If it is take 1 unit of DDT, then such 10 fishes taken by larger fish, then 10 times the concentration will increase. Then another larger fish, if it takes this fish, then 100 times the concentration will increase. Then if a human consume this fish, then 10 fishes if he consumes, 1,000 times the concentration of the DDT will increase in the body of human, and it will settle in the body itself. It will not be released or removed from the body, because the body cannot metabolize the DDT. So DDT will accumulate in the body slowly in the biological system, not only in human. So the DDT will accumulate slowly in the biological system, and finally it will give its disastrous impact to the biological system. That was first came to light through the writings of Rachel Carson. Then after this release of this book, every country started to ban the use of DDT, including India. India banned the use of DDT for agriculture purpose. But still India is using DDT in Bihar to control sandflies. Once the malaria elimination program was started in India in 1960s, so people were indiscriminately used DDT to kill the malaria-causing, malaria-transmitting mosquitoes. But at one stage, they were all very happy. The program people were happy that they are at the point of eliminating malaria from India. So almost they have eliminated mosquito. Almost they have brought down the cases to zero, malaria cases to zero at the end of 60s. So they started to wind up this program, malaria elimination program from India. But to their surprise, it has come again like tsunami. Malaria has come again like tsunami, and it was attacking people and it was killing people like never before. But after stopping this spraying insecticide, after stopping this program, it has come with new vigor, with a lot of force. It has come and it has attacked the people of India, and lot of people, the unimaginable number of people were affected by this malaria. Then again government restarted the malaria elimination program, but this time they have not used DDT. They have used alternative chemicals to control this malaria transmitting mosquitoes. After that, due to the malaria elimination program, lot of unwanted things happened, like accumulation of DDT, so many things. But one favorable thing also happened is the purpose of spraying DDT at the time was to eliminate malaria transmitting mosquitoes. But as a side effect, as a side benefit, these DDT also killed sand flies. Sand flies are the insects which are smaller than mosquito. They are transmitting disease to human that disease is called leishmaniasis. Leishmaniasis was once very vibrant in Tamil Nadu also. A lot of people died in Chennai, and Leishman Donovan was doing research in Chennai only, and Donovan was doing research in Calcutta, and Leishman was doing research in Leishmania parasite in Chennai during British period. So after that, due to the well infrastructure of Tamil Nadu public health system, now the leishmaniasis is under control, or we can say that it has been totally eliminated from Tamil Nadu. But even though now it is reappearing in the border districts of Tamil Nadu. But it was a very dangerous disease in Bihar, West Bengal, and eastern Uttar Pradesh. So in those areas, the leishmaniasis are very popular, and government is trying, government is making so many attempts like elimination program by 2010. It was the program started by government during 2008 or 7. So they were aiming to eliminate this leishmaniasis from the world within 2010. Ana government ala mudiyala, adu eliminate panna mudiyala. So they have again extended the period 2015, 2020. So now they have extended the period to 2025. Now they have changed all the definitions, like if there is initially they have formed zero cases of leishmaniasis is the indication for removal of leishmaniasis, but later they have changed the definition, like in 1 kilometer area, if one case is there, we can consider it as eliminated. Likewise, they have converted the definition also now, but really the government is spending a lot to get success in elimination of leishmaniasis. But what I want to say here is, as a side effect of malaria elimination program, the sandflies were eliminated from those area, from the affected area. So the leishmania cases started come down at that period. So as a side benefit, people enjoyed this. But now again, after stopping this malaria elimination program, again the leishmaniasis cases went up, and now government is allowing to use DDT for elimination of leishmania program, even though other alternative chemical methods available. Since mosquito developing resistance to insecticides, people are always increasing the concentration. Say for example, if 1 kilo of DDT, for example, if it is sprayed in 1 acre area, that is not acceptable for mosquito, then people started to increase 2 kilo per acre. Then they started to increase 10 kilo per acre. In that way, slowly people started to increase the concentration, but beyond certain level of increasing the concentration, that will be very much harmful to the people. So increasing concentration is not an advisable one. Then come to insects. Why we are talking about insects? So we have to know about insects, because insects are the vectors of so many diseases, beneficial insects available. So we have to know about insects. Insects appeared late Carboniferous period, some 300 million years before, and 8,50,000 species known so far, and every year several thousands of new species are discovered, and it is added to the list. And say nearly 80 to 95% of the insects are yet to be collected and named. So what we know is only 5 to 15%. We know 5 to 20% of insects only we know, but remaining lot of things yet to be discovered, and it's all the responsibility of you younger people to discover the undiscovered insects in the field. Definitely, if you go to Western Ghats, if you're looking for some insects, definitely you will get some new type of insects. So you can find, you can describe the insect, and you can publish the data in journal. And if you are the first person, then they will give your name also. Say, for example, your name is Subramani, then Subramaniam they will give in the name to the insect. So it is a vibrant area, you can try. And very little information is available about the feeding habitats, what the insect will feed, because every insect is an individual one. They are different from each other. If you take mosquitoes, some mosquitoes are there, they will take blood only from human. Some mosquitoes will take blood from both human and animal. Some mosquito will take only honey. It will not take blood. It will take only honey. So just you see in the mosquito itself, there are wide variety of feeding habitats. So if you imagine, if you try to understand the whole insect, then you will get lot of novel things, lot of novel feeding habitats, how they feed, what they feed, how they digest. Because if you take the Karayan Riklapa, termites. The termite's stomach is a very beautiful area where the microbes in the termite gut produces methane. People are now trying to understand the mechanism which is going on in the termite guts to use wood material to produce fuel. So it may be the alternative fuel technology for future. So in that way, every insect is unique. So we have to try to find out what type of mechanism is going on in collecting the food, in digesting those food material. But only one property is shared by all the insects, that is their association with microorganisms. Insects are sharing the habitat with microorganisms. Microbiology students இருக்கீங்களா பாங்க? Or all zoology. Okay. So the only character the insects are sharing with other is its association with the microorganism. No insect can be successful without microorganisms. So microbes. If we talk about microbes, microbes are the only group on the earth that can surpass insects in diversity. Microbes எடுத்துக்கிட்டீங்கன்னா still it is diverse. பாக்டீரியாவும் சேர்க்கலாம், ஃபங்கஸ் சேர்க்கலாம், ஆல்கே சேர்க்கலாம். ஒவ்வொன்றிலையும் பாக்டீரியாலையும் you can culture only 5%, but remaining 95% you cannot culture even. So it cannot be cultured. If you are trying to get soil, 1 gram of soil, and if you are trying to isolate bacteria from 1 gram of soil, then you will get hundreds of colonies, different bacterial colonies on the culture plate. But that is very limited number. Only 5% you can make them to grow. Remaining 95% you cannot make them to grow because we don't have facility to provide nutrient to the remaining 95% of bacteria. So you can identify the presence by genomic techniques. By metagenomic analysis, you can find out the presence of microbial diversity in any particular sample. After the origin of Earth, nearly 3.8 billion years, these organisms formulated the Earth. So, Earth formed first, and there were no living organisms on the surface of the Earth for several billions of years. Earthல ஒரே அப்படியே எரிமலை வெடிச்சுக்கிட்டு புயல் அடிச்சுக்கிட்டு இப்படி தான் கிடந்துச்சு. But after several billion years, Then microbes started appearing on the surface of earth. That was the first living organism on the earth. So first microorganisms appear, then adula diversify agudhu. Different groups started appearing. They started to harvest the energy from sunlight, and they started to harvest fixed atmospheric carbon dioxide, and they started to produce carbohydrate they started to produce. That was the basic for the formation of life afterwards. So after the first bacteria appeared on the earth, one by one, lot of organisms slowly started emerging with a gap of some million years, some several thousand years, million years after, with a gap of those things, several animals started appearing on the earth. For nearly 2 billion years, these organisms were the only life on earth. They formulated the basic biogeochemical cycles of the earth. So first microbes appear ஆயிடுச்சு எர்த்தில். அது வந்து எர்த்தை ரெடி பண்ணுது. Other organisms, other living animals அங்கே உருவாகிறதுக்கு they are making earth to be suitable for other higher animals. So எர்த்தை வந்து அது ரெடி பண்ணுது. So earth வந்து ரொம்ப ஹார்ஷாக இருக்குது. That harsh earth, newly formed harsh earth, was pacified by microorganisms. அதை ஸ்மூத் பண்ணி make that microorganisms made the earth suitable for the survival of higher animals. So அதனால என்ன ஆகுது, ஒன்னொன்னா other animals started appearing on the earth. Finally we appeared. So if you take the geological time scale as a 1 year period, so just imagine, consider the whole period of earth formation to the present day, you consider it as a 1 year period. Okay? Human, when human appeared December 31st, nearly 11:55. If you consider the whole period as of 1 year, human appeared only in the December 31st night, 11:55 a.m. So only he passed 5 minutes of 1 year period. So he passed only 5 minutes of the whole geological time scale period. He passed only 5 minutes. Other organisms passed so many period before us. They are so much ancestors to us. Insects, why we are afraid of insects? Because they are all ancestors to us. They are very knowledgeable to us. We have large brain, but that itself not an indication of knowledge. Even if you take elephant, which is having brain larger than human, but the ratio of brain, how much the brain is used for their survival, that is important. So killing insect is very not easy, because they have lived on the earth several million years back to us. So they know very well about the earth, about the conditions, about the temperature, about the pH of the earth, and they live successfully so much period. We only jujube. நம்ம ஜஸ்ட் இப்போ தான் அப்பியர் ஆகும். உங்கள் ஜூனியர்ஸ் உள்ளே வந்தாங்கன்னா எப்படி ட்ரீட் பண்ணுவீங்க அவங்கள? நீங்களாம் சீனியர்ஸ் உள்ளே இருக்கும்போது ஒரு ஜூனியர்ஸ் உள்ளே வந்தாங்கன்னா ஜஸ்ட் அவன் இப்போ தான் அட்மிஷன் ஆகிட்டு உள்ளே நுழைஞ்சிருக்கான். ஸோ வி ஆர் லைக் தேட். ஸோ ஹியூமன் பீயிங்ஸ் ஆர் ஜஸ்ட் ஜூனியர்ஸ் டு ஆல் தி அனிமல்ஸ் லிவிங் ஆன் தி வேர்ல்ட். ஸோ we should not just take it granted to disturb other animals. How microbes are essential for the insects? They are required for symbiotic relationship. Lot of microbes living, in research we are finding, or we are in the process of finding, the role of microorganisms in the gut of mosquito. The microorganisms in the mosquito midgut are responsible for controlling the mating behavior. If you alter the microorganisms of the midgut, the mosquitoes are not mating properly. Say, for example, if you are treating mosquito with some antibiotic, tetracycline if you are giving, and after that you are allowing the mosquito to mate, it will not mate normally. So it's all indication that microbes providing everything to the living animal. Even lot of research is going on in human. Our human habitat is determining what type of microbes we are harboring, and habit is determined by the microorganisms. So somebody may be very fond of sweet, somebody may be fond of karam. காரங்களை விரும்பி சாப்பிடலாம் சிலர், ஸ்வீட் விரும்பி சாப்பிடலாம். Somebody may be fond of non-veg. So these things itself determined by the gut flora of human. இதெல்லாம் latest findings in nature, science, இந்த மாதிரி ஜேர்னல்ஸ் எடுத்து பார்த்தீங்கன்னா மைக்ரோபயோட்டா, கட் மைக்ரோபயோட்டா ஸ்டடீஸில் ஹியூமனில் நடந்துக்கிட்டு இருக்க ரிசர்ச் எல்லாமே என்ன சொல்லுது அப்படின்னா, ஈவன் ஹியூமன் பிஹேவியர் இஸ் கண்ட்ரோல்டு பை மைக்ரோப்ஸ். Because if you count the number of cells on our body, in our body, if you count, and if you count the number of bacteria which is living inside our body, is far greater than our own cell. Say, for example, if my body is made up of 10 million cells, for example, bacteria we are harboring is 100 million. So who is mass here? Bacteria is greater than us. Bacteria which are living inside our body is much, much, much greater than our own body. So your body mass is, when you take mass, your body mass, the entire body mass, say for example you are 50 kg, the bacteria comes for around 2-3 kgs. You may not think that it is, by weight it is less than human. No, by size it is greater than human, because bacterial size is very small. So one cell of human cell is equal to hundreds of bacteria. So the number is very high. If you take the weight, every human may harbor 1 kg, 2 kg, 3 kg microorganisms inside our body. So they are determining our ability. If you are today, if you are feeling happy, means you are having healthy microbiota. If you are feeling sad, then there will be some disturbance in the microbiota. So you are feeling sad. So every behavior also controlled by microorganisms. So lot of avenue is there opened for you to research. So microbes control everything, and we are doing little bit in that. In our research center we are trying to study the germ-free mosquitoes and how mosquito behaves if they are living without any microbes. That we are calling it as germ-free mosquitoes. So how germ-free mosquitoes are living, and how mosquitoes behaving after giving one type of microorganisms, after giving two type of microorganisms, that we are trying to understand. So this is geological time scale I have just described. Human just appear only very late. Back to mosquitoes. As I told you, microbes control everything. In mosquito also, microbes controlling everything, starting from the eggs. Mosquito, we cannot just think about mosquito as a simple insect. Mosquito, after taking blood meal and after mating with the male, mosquito will not lay her eggs on the way wherever water bodies are available. போகிற போக்கில் அது வந்து எக்லே பண்ணிட்டு போயிடாது. It finds very meticulously the suitable habitat for egg laying. அப்படியே பறந்து போய் it will sense, the mosquito will sense the water body, whether the water body is suitable for the young one. So அது மாதிரி அப்படியே it will sense the water body. So after confirming that the water body is suitable for her young generation, then the mosquito will lay eggs in those water body. So first it will sense, that we are calling it as ovipositioning. So ovipositioning is a very long study in mosquito biology. So mosquito identifies the breeding place very meticulously and lay her eggs after finding the water body suitable for growing young ones. So after laying eggs, lot of things going on in the water body itself. So the mother identified the place is suitable for young one. How it identifies? By the emanation of scents, smell released from the water body. So that may be of different microorganisms, may be there in the water body, different type of organisms, protozoans may be in the water body. Those things are required for the growth of mosquito. So after identifying the presence of those things, the mosquito lay her eggs in the water body. So then after laying eggs, young one comes out from the eggs and immediately the food is ready for mosquito larvae to eat. Time kuda podo. So lot of food web is going on wherever the mosquito lays eggs. And it also, mosquito also finds which human to bite. So that is called scent emanation from the skin surface. Humans are attractant to mosquitoes. Somebody's are more attractant, somebody's are not that much attractant. Everything is determined by the microbiota of skin. So that is a factor that determines what type of mosquito biting you and how much it is biting you. So modern vector control methods, Bt toxins, sterile insect techniques, genetically modified mosquitoes, Wolbachia endobacteria, RNAi, entomopathogenic bacteria, fungi, insecticide impregnated bed nets, and electric devices. So Bt toxin, which is still used biological control method, successfully used method to control mosquitoes, released by the toxin released by Bacillus thuringiensis. Now scientists are trying to develop friendly mosquitoes. So if they release the friendly mosquito, it will mate with the female and no progeny will be produced. Those mosquitoes, we are calling it as friendly mosquitoes. That can be achieved by sterile insect technique. In that, lot of gamma irradiation will be given to male mosquito, and male mosquito genetic thing will made impotent. They will be made impotent or sterile. It will not make female mosquito to produce eggs. So this type of techniques people are trying, but it is not so successful because the irradiated mosquito are not that much active with the wild mosquito, wild male. The irradiated male are very dull and they are only performing poor comparing with wild male mosquito. So this method is not so successful. And genetically modified mosquitoes, people are trying to change the sequence, to do some alteration in the gene sequences to make the mosquito such a way that some control mechanism is there in the gene. So if one protein is overexpressed, then the protein expression will be stopped by activating the stop codon. So now people are trying to use this mechanism to stop the production of vital protein for the mosquito itself. So mosquito will not survive. If you are incorporating the fluorescent gene with those incorporated gene, then you can easily identify the genetically transferred mosquitoes. It is an indicator. People are releasing the modified mosquitoes. Wolbachia, we are working with that Wolbachia bacteria. Wolbachia is endobacteria. The Wolbachia means, endobacteria means it lives inside the cell, like virus. Virus, as we know, it is intracellular parasite, lives inside the cell. Like this Wolbachia also, intracellular parasite bacteria, which lives inside the cell. So if you are releasing the Wolbachia infected male and female, it will not survive in the environment for long because no progeny will be produced based on this method. But one thing, The Wolbachia, if it is taken from the mosquito itself, it will not work. So we have to take Wolbachia from fruit flies. So the Wolbachia taken from fruit flies works very well with mosquitoes. Mosquitoes are also harboring Wolbachia, but that Wolbachia will not give any disturbance, any mechanism change in the mosquito. But it's a very tedious process. Even if you are trying to introduce the Wolbachia from other species into mosquitoes, you have to make several attempts. Say, for example, if you are making 1,000 attempts, only one success you will get. So now they are releasing the Wolbachia infected mosquitoes to the field, and now we are also trying with the nanotechnology to encapsulate the bacteria that can produce mosquito larvicides in nanofiber. Generally, what happens if you are releasing this type of bacteria without any protective cover, this bacteria cannot survive long in the environment. It will be killed immediately by the environmental conditions. So to avoid that, now we are trying to encapsulate the bacteria. This photograph is taken by me. We are encapsulating the bacteria with nanofiber. Then after that, if you are releasing this nanofiber encapsulated bacteria in the field, then the bacteria happily survive inside and releases the mosquito larvicide to the environment for the larval control. And entomopathogenic fungi is also another method, and my officer in charge, Dr. Paramasivam, is working with this. He is trying to produce fungal pathogens that can infect mosquitoes. This is RNAi technique, RNA interference technique. This is simply to stop the function of RNA that behaves normally. So by the way of doing this, we can stop the function of metabolic activity of insects. And this is very old method, using fishes in the field to consume larvae. But mosquito, as I told you, mosquito very intelligent. If you are introducing a fish in the pond, then it will not lay her eggs in the pond. Any time if you are releasing fish, then mosquito will choose another place to lay her eggs, because mosquito knows that the water body contains fishes. So this method is not so successful one. Dragonfly is a natural predator, and one of my colleagues is working with this dragonfly insects. They are the natural controller of mosquito larvae. In the water body, dragonfly larvae feeds on mosquito larvae, and adult dragonfly feeds on adult mosquitoes. So natural control mechanism, these are all. This is olfactometer experiment. This is mosquito receptor experiment. But still, using bed net is the good method, very good method, better method, best method I can say. So you can avoid mosquito biting by using nets in your windows, house, and you can, if it is not possible to make your house mosquito proof, then you can use mosquito bed nets to avoid mosquito biting. So recently these devices of Chinese made make available in India to kill mosquitoes, but they are also working well. You can locally, to remove the mosquitoes, you can use these devices. Thank you so much. If you have any doubt, you can ask me. Still lot of things I want to talk, but due to the shortage of time, we can meet some other time. Yeah. RNAi technique, RNA inhibition technique it is called. So generally what happens, RNA produced from DNA, that is the translation method. By translation, proteins are synthesized from RNA. By translation, RNA is the base. From DNA, RNA are produced, and from RNA, proteins are produced. So DNA is transferring its information through RNA to protein. You can stop at that level. You can use some inhibitor to stop functioning the RNA. So by doing so, you can prevent the some metabolic activity, selected metabolic activity. No, it is in research level only. Now we are using Wolbachia, that is in field stage. Yeah, to make mosquitoes not transmitting diseases, dengue like.
Any other questions from the student side?
இது வந்து ராங் நோஷன் பா. அதாவது ஹீட்டான பாடின்னு எதுவுமே கிடையாது. ஹியூமன் பாடி டெம்பரேச்சர் இஸ் கான்ஸ்டன்ட். ஹையா எப்படி இருக்கும்? அவர் பாடி டெம்பரேச்சர் இஸ் கான்ஸ்டன்ட். No, let me clear you. Human body temperature is constant one. Okay, our body maintains constant temperature irrespective of environmental temperature. வெளியில கோல்டாக இருக்கும், but your body maintains 37 point something temperature to keep your body active. We are in a two type of organisms, cold blooded, warm blooded animal. We are warm blooded animal. We used to keep the temperature always constant. So there is no question of cold body, hot body. We are all always hot. But mosquito choose the human being based on the scent released by the each individual. Okay. Okay. Maybe some other mechanism may be in that, not exactly the temperature. Okay.
Any other questions?
சொல்லுங்கம்மா.
How much is efficiency for controlling the mosquitoes?
But these days people are using silver nanoparticles because that is comparatively cheap when using gold nanoparticles. You can also use gold nanoparticles, but people are using silver nanoparticle. Some experiment, some of my colleagues are doing with the silver nanoparticle, but my experiment is different. I am doing with the nanofibers. Yeah, that is using creating a biopolymer. So I am encapsulating the bacteria with the biopolymer. It's like a fiber, nanofiber, not the particles.
How much is efficiency cost for?
Control
the microbes.
Yeah, there is no change in efficiency. I told you, microbes are producing larvicides, but those microbes cannot survive long in the field because due to the impact of field conditions. So we have to give some protective cover to the bacteria. Bacteria produces as usual the larvicides. So we are giving extra cover to the bacteria. Yeah, it is possible because Wolbachia related with cytoplasm. That is why in my slide I have shown it prefers female because it allows only female to survive. It will not allow male to survive because it is transmitted through cytoplasm. I have told you that Wolbachia is an intracellular bacteria. It lives inside the cell. So if it is living inside the egg cell, if you take egg cell, then it can pass through the next generation. If it lives inside the sperm cell, it cannot transfer to the another generation because after mating with the egg, only nucleus will go inside the egg, the remaining everything will be go away, will be washed away. So this Wolbachia bacteria prefers only female to survive. It will not allow male to survive. because it is living in the cytoplasm, eggs only will be passed to next generation. So the cytoplasmic things will be transferred to the next generation.
What is the inoculation technique, sir? Like how
Microinjection. By careful microinjection, you can inject the Wolbachia into the mosquito eggs. By inserting the syringe needle into that, you can push, which are all computer controlled. You can just, you should learn the technique how to introduce this into the eggs. That's all.
Thank you.
Okay, thank you so much.
A few minutes. So it is our duty to express our sincere thanks to the speaker of the session, Dr. Muniraj. So it was very useful to us, and our students are exposed to the not only the vector control, but also the history behind the science and technology development in India. So we express our sincere thanks to Dr. Muniraj. We also express our thanks to Dr. Victor, who accompanied him. So thank you all for your patient hearing. Thank you. Thank you, one and all.
If you want any help from us, from VCRC, you can come to us. You can get mosquitoes, if you want to do any research work, project work, you can come to us.
The tea has been arranged at the back side of the department. Please do go have it.